Analyzing the content of styrofoam consuming superworms waste by Sarah and Ashley.
So in our experiment, we're working with superworms. They're scientifically known as Phobos morio, and they're the larvae of darkling beetles. We're working with them because they have a bacterial enzyme within their gut that breaks down polystyrene, also known as styrofoam. So we trained a group of them to have a diet based on only styrofoam.
Polystyrene degrades slowly on its own, and it contains harmful chemicals that leach into the environment, and it contributes to global warming that 29 million tons that are produced annually, and it's commonly used for delivery packaging, foam cups, coffee cups, and to-go containers.
Our question is, when styrofoams on a styrofoam-based diet excrete waste, is there toxic material in their waste being recycled back into the environment?
Our previous experiment that we did last year contained of a tomato plant with normal soil, that was our constant, versus tomato plant with normal soil that contained the superworms waste containing the styrofoam. And we came to a conclusion that while the tomato plant with the normal soil was growing, the other tomato plant containing the waste was dead less than a week. We concluded that the waste containing the styrofoam still contained toxic chemicals that contaminated the soil, causing the plant to die.
This year's experiment, our control plants are chicory plants grown with pure superworm waste in the soil, and our experimental plants are also chicory plants, but their soil has the waste of styrofoam diet superworms in it.
What are we testing? We're comparing the growth of the experimental plant to the growth of the control plant.
For our materials, we're using chicory seeds,
Superworms,
styrofoam.
and superworm tank.
For our procedure, we water our chicory plants 5 days a week.
We have styrofoam that we resupply to our superworms with food 2 times a week.
And we're going to start taking waste samples once every 2 weeks. Our hypothesis is if the experimental plant's growth is lower compared to the control plant's growth, we can conclude that the toxic material from the styrofoam is what's causing the plants to die, and not the waste itself.
What are methanogens, you might ask? Methanogens are methane-producing bacterium, especially in archaean, which reduces carbon dioxide to methane.
We plan to use methanogens to neutralize any toxins we find from the worm's waste.
Results we're expecting to see after our experiment, superworms will be able to biodegrade styrofoam, methanogens will neutralize any remaining toxins in the waste, and experimental chicories will be replanted, and the styrofoam consuming superworms waste will not affect its growth. Thank you.
This is Fish vs. Microplastics by Ashley Budeman.
And Isaac Otzperu.
So the problem is that plastics in our oceans are increasing rapidly. Many organisms have been taking plastics, which can be whole bags or even small beads, but it slowly affects their system and kills them. The plastic then gets passed on organism to organism and slowly accumulates. So here's some statistics. It's estimated that there is 5.25 trillion pieces of plastic in the ocean. Plastic kills over 1 million animals per year, and 100,000 of these organisms die from being entangled. Plastic makes up for 80% of ocean's pollution, and 300 million tons of plastic get created yearly, and only 50% is single use. Here are some photos, and as you can see in the top, there's the Great Pacific Garbage Patch, which is the largest trash site on the planet. It's located in the North Pacific subtropical gyre. So here's my reasoning for this experiment. We have changing aspects of the ocean wildlife, which is due to the uprising pollution. Plastic is reducing our biodiversity. Many organisms are basically becoming endangered, such as the Hawaiian monk seal. And we also want to personally see and show the effects of plastic on fish. Like I said, microplastics in animals are passed on to the next organism that consumes them. Okay. Our question, how would the weight and quantity of offspring alter if microplastics are introduced into the Danio Rerio diet? Here's some of our research. Some of the researchers found that the fish that ate plastic gained weight and the females did not, and then others found out that the females had less production of eggs. Our hypotheses. According to our research, we can hypothesize that if the fish in tank B are consuming microplastics on a regular basis, they will weigh much less than the fish in tank A. We can also hypothesize that the fish in tank B will have a smaller quantity of offspring compared to tank A. Here are our materials. their basic fish keeping stuff, and our 2 tanks. The tank A is our control, which will have no plastic, and tank B is our experimental, which will have plastic. For this experiment, we're using zebrafish, which we will be feeding 1 time a day. Like I said earlier, tank A will have normal fish food, and tank B will have the food with the microplastics. We're using these fish, which are also called zebrafish or Danio. They lay eggs, and their embryos are almost completely translucent. Almost all fish used in medical research are zebrafish because of their translucent qualities and because they're so easily manipulated. They can live 1 to 3 years in a fish tank. To make the microplastics, we're using high-density polyethylene, which we got from common items such as shampoo bottles and milk containers. Our procedure is basically buying the materials, setting up our tanks, and continuing with our data by feeding the fish the plastic, and then weighing them every 2 weeks. We'll be weighing them by putting them on a scale and then calculating the average weight for each tank. And to breeding them, we will be breeding each pairs from each tank and counting the number of offspring. There's our references. Thank you.
Our presentation is the analysis of the effects of ocean acidification on Helisoma trivolvis by Eliza Oy.
And Trey DeMaclonan.
Oceans absorb about 30% of the CO2 in the atmosphere, and this process is known as ocean acidification. Ocean acidification, in broader terms, is the reduction in the pH of the ocean. The cause of ocean acidification is primarily the combustion of hydrocarbons, such as burning fossil fuels and land use change. Food webs, carbonate-based shellfish, and coral could be negatively impacted by ocean acidification.
So the organism that we're testing our project on is Helisoma trivolvis, and they're mostly found in lakes, ponds, swamps, and riverine backwaters. They thrive in rich eutrophic environments, which means they thrive in a lot of nutrient-rich waters, and they lay 20 to 40 eggs per week, depending on the culture conditions. So the purpose of this project is to find an efficient way to sequester carbon in the ocean. And we're trying to reduce the amount of carbon dioxide pollution in our environment while reducing the effects of ocean acidification on aquatic life. So our question is, how do the effects of carbon dioxide diffusion on ocean acidification impact Helisoma trivolvis and their ability to adapt to acidity?
So our materials include a tank with fresh water, baking soda, and carbon dioxide tests, Helisoma trivolvis, which is our organism, snail food so that we could feed our organisms, and Elodia. So our procedure is we are first going to breed the Helisoma trivolvis, which is what we are currently working on now, and we're going to fill our tank with fresh water and add CO2 into the tank, which is done by baking soda, and then later we will test how much CO2 is actually in the tank with carbon dioxide tests. Then we're going to observe the short-term and long-term effects of the CO2 diffusion on the Helisoma trivolvis, and then later we will replicate the same procedure with salt water.
So future plans for this project are that we're going to replicate this experiment in saltwater, and we're going to test it with different variables since the conditions for freshwater and saltwater are kind of different. So we're going to use saltwater plants, organisms, and we're going to do the conditions that are more suited for saltwater. And we're probably going to follow the previous seniors projects, which is like a continuation of what they did before. And that's all.